Dengue IgM capture ELISA. Introduction. Dengue is a mosquito-borne viral disease and may cause a subclinical infection or a severe flu-like illness and could occasionally be fatal. Example: dengue hemorrhagic fever or dengue shock syndrome. Dengue is transmitted by several species of mosquito within the genus Aedes, principally Aedes aegypti. Principle of assay. IgM antibodies in the patient's serum are captured by anti-human IgM coated onto the solid surface wells. In the next step, dengue antigen is added, which binds to captured human IgM in the sample. Unbound antigen is removed during the washing step. In the subsequent step, anti-dengue monoclonal antibodies added, followed by avidine histidine-rich protein. Subsequently, chromogenic substrate is added and the reaction is stopped by sulfuric acid. The intensity of color or optical density is measured at 450 nanometers. Reagents provided in each kit. The dengue IgM ELISA kit used in this demonstration contains the following: anti-human IgM-coated wells. These are ready to use. 12 strips with 8 wells each are coated with anti-human IgM antibodies. This is stable at 2 to 8 degrees centigrade if protected from moisture. Sample diluent for dengue IgM, ready to use. Phosphate buffered saline with additives and antibiotics for dilution of test samples. Wash buffer concentrate. Before use, dilute wash buffer concentrate 1:19, that is, 1 part of buffer concentrate and 19 parts of high-grade distilled water. For assay of 8 samples, that is, 6 clinical samples and 2 controls, 100 milliliters of diluted wash buffer is sufficient. Dengue antigen, ready to use. Anti-dengue monoclonal antibody, ready to use. Avidin HRP, ready to use. Protect the solution from direct exposure to light. Liquid TMB substrate, ready to use. Protect this from light. Stop solution, ready to use. This solution contains 1 normal sulfuric acid. Wear protective gloves, mask and eyeglasses while handling stop solution. Dengue IgM positive control, ready to use. Dengue IgM negative control, ready to use. This will help to monitor the integrity of the test kit. Storage conditions. It is essential that all reagents and material is stored at 2 to 8 degrees centigrade. Do not freeze any of the reagents. Do not bring or equilibrate any of the reagents to room temperature even on the day of performing the test. Anti-human IgM coated test strips must be protected from moisture. The unused strips should be placed safely in self-sealing pouch with desiccant. Additional material required: microplate washer, microplate reader with 450 nanometers filter, incubator at 37 degrees centigrade, adjustable micropipette with disposable tips, high-grade glass distilled water, tissue paper or aluminium foil, glass tubes for dilution of sample, timer. Preparation of reagents before performing test. Dilute wash buffer concentrate by mixing 1 part of wash buffer concentrate with 19 parts of high-grade glass distilled water. For assay of 8 samples, that is, 6 clinical samples and 2 controls, 100 milliliters of diluted wash buffer is sufficient. The unused diluted wash buffer can be stored in a refrigerator for 2 months or until it shows microbial growth. Remove the anti-human IgM coated strip as per the number of samples to be tested and fix it on the strip holder. The unused strip must be sealed immediately with the film and kept in the aluminium pouch. The unused strips must be protected from moisture. At the time of addition, take the reagents from the refrigerator. Add required quantity of the reagent to the wells and keep the vial back into the refrigerator. Do not bring any of the reagents to room temperature. This may lower the shelf life of the kit. After washing the strips, the next reagents must be added immediately before the wells become dry. Dried wells may give erroneous results. Test protocol. Thoroughly mix all the reagents using the micropipette tips before addition. The test procedure must be followed meticulously. Don't bring any of the kit reagents to room temperature before commencing the test. All reagents are stable at 2 to 8 degrees centigrade and for in vitro use only. Procedure. Select the samples to be assayed. Write down the protocol on ELISA sheet provided with each kit. Dilute serum 1 in 100 in tubes or preferably in deep well plate using sample diluent for dengue IgM. Remove required number of anti-IgM coated strips. Number the test strips as 1, 2, 3 and so on. Wash the strips 3 times with wash buffer. Do not allow the wells to dry. Transfer 50 microliters of diluted samples from the deep well plate to respective wells using multi-channel pipette. Add 50 microliters of dengue IgM positive control and dengue IgM negative control to respective wells as per the protocol. Cover the plate with aluminium foil to prevent evaporation of samples. Keep the plate in a closed humidified box inside the incubator and incubate the plate at 37 degrees centigrade for 1 hour. In our demonstration, the ELISA washer has an inbuilt chamber for incubation. Use timer for accurate incubation period. At the end of incubation, wash the plate 5 times with wash buffer. Tap the plate after last wash on a tissue paper to remove traces of wash buffer content. Add 50 microliters of dengue antigen to each well of the plate. Cover the plate with aluminium foil to prevent evaporation of samples and incubate for 1 hour at 37 degrees centigrade. At the end of incubation, Wash the plate 5 times with wash buffer. Tap the plate after last wash on a tissue paper to remove traces of wash buffer content. Add 50 microliters of anti-dengue monoclonal antibody to each well. Cover the plate with aluminium foil to prevent evaporation of samples and incubate for 1 hour at 37 degrees centigrade. At the end of incubation, wash the plate 5 times with wash buffer. Tap the plate after last wash on a tissue paper to remove traces of wash buffer content. Add 50 microliters of Avadine HRP to each well. Cover the plate with aluminium foil to prevent evaporation of samples and incubate for 30 minutes at 37 degrees centigrade. At the end of incubation, wash the plate 5 times with wash buffer. Tap the plate after last wash on a tissue paper to remove traces of wash buffer content. Add 100 microliters of liquid TMB substrate to each well. Incubate at room temperature in dark for 10 minutes. Stop the reaction exactly after 10 minutes with 100 microliters of stop solution. Measure the absorbance at 450 nanometers within 10 minutes after termination of reaction.